Welcome to today's lecture. Today we are going to talk about humoral immune response. Humoral immune response. The B cells are activated and produce the antibody for elimination of the antigen. Because most of the antigen belongs to the thymus-dependent antigen, TD antigen. That means it requires the involvement of T cells to activate B lymphocyte. So today we mainly focus on TD antigen-mediated primary humoral immune response. When antigen invade into our body, they will be carried to the peripheral lymphoid organs, such as the spleen, the lymph node. In the peripheral lymphoid organs, they will meet with the B cells and T cells. So B cells and T cells, they will be activated and interact with each other. Finally, the B lymphocyte will grow into the plasma cells for the production of the antibody. And then antibody will spread all of the body to eliminate the antigens. This is the general process of the humoral immune response. But here, I would like to emphasize that the response takes place in the peripheral lymphoid organs instead of where the antigen entered. So today, we will take the lymph node as an example to explain how the primary humoral immune response takes place. So antigen will carry it to the lymph node through the afferent lymphatics or from the blood. The B lymphocyte coming from the blood, the B cells from the blood exist in the follicles. And how the B cells can see the antigens? In the subcapsular sinus, there are many macrophages. Those macrophages can bind with antigen efficiently. They will not internalize the antigen. They just keep the antigen on the surface. So the accumulation of the antigen by the macrophage are very important for B cells to sample the antigens. There was another cells. In medulla, there are some dendritic cells. They are also very important for B cells to sample the antigens. So once the B cells recognize the antigen, but how the B cells are activated? The activation of the B cells requires the involvement of the T cells. So T cells and B cells, they must interact with each other, and then the B cells can be activated to produce the antibody. And here I would like to emphasize that B cells and T cells exist in different locations in the lymph node. B cells are usually in the follicles, and T cells in the T cell zones. So B cells and T cells, they must move towards each other. So after sample the antigens, the B cells will migrate towards the T cells. At the border of the B cell zones and the T cell zones, they interact with each other. And in another, B cells and T cells, they must recognize the same antigen. But B cells and T cells see antigens in different way. B cells can recognize the antigen in a native form. For example, this is a bacteria, and BCR recognize the antigen on the surface of the bacteria. But the T cells cannot recognize the antigen on the surface of the bacteria. They must recognize the MHC molecule presented antigen. So when the B cells bind with the antigen, they will internalize those antigens inside of the cells and then degrade them into small pieces. The small peptide will bind with MHC molecule. They form a complex to express on the surface of the B cells. Then the T cells can recognize the MHC molecule presented antigen. So this will give the T cells the first activation signal. But both T cells and B cells, the activation of both cells required at least 2 signals. They recognize the antigen just to provide the first signal. B cells express CD80 and CD86 molecule. T cell express CD28 molecule. These 2 molecules can bind with each other, and their binding will release a secondary signal for T cells activation. So receive both signals, T cell is activated. After T cell activated, the activated T cell will express CD40L to bind CD40 on the surface of the B lymphocyte, which will give a secondary signal for activation of the B lymphocyte. So this recognition is called linked recognition. In addition, after the T cell activated, they will release some cytokines, Th2 cytokines, L4, L5, and L6. They will promote activation of the B lymphocyte. Of course, the B cells and T cells, they will express some other molecules to sticky themselves very well to ensure T cells and B cells, they can interact with each other tightly. After B cell is activated, they will proliferate to form the primary focus, and where they will grow into the plasma cells and for the production of the antibody. But here, the plasma cells are short-lived, and the antibody produced are mainly IgM. And this antibody contributes the first phase of the primary humoral immune response. While other activated cells will migrate into the follicles, as shown in the yellow. So other B cells migrate into the follicles to proliferate, to form the germinal center. Here is the germinal center. And in the germinal center, B cells proliferate, and eventually they will differentiate into the plasma cells and memory B cells. The plasma cells are long-lived, and they will leave the lymph node to migrate to the bone marrow for antibody production. But here, the antibody includes all kinds of the antibodies, IgG, IgM, etc. That is the second phase of the primary humoral immune response. And small amount of the activated cells will grow into the memory B cells. Memory B cells can remember what kind of the antigen they have met. So once again they met with the same antigen, they will elicit a very quick response to eliminate antigens. So that is the secondary immune response. That's all for today. Thank you for your attention.